Hello guys, this video is supposed to help you understand the homework for tonight, which is to complete parts 3 and 4 of the Meiosis modeling activity. So this video is going to have two parts. The first part is just a review of what we did today and a description of why it's important, what I'm meaning to get at in this activity. And the second is going to be just a demonstration of how to do the homework for tonight. All right, so before we start, I want to thank Donald for taking a picture of himself on my phone and then hiding it, so I couldn't find it for a half hour. Thanks a lot, Donald. That was really helpful. Yeah, let's get going now. Okay, so we've got a diploid cell here, and we can tell because there are two versions of each gene, right? So we've got a B star here and a B here, E star here, E here. These are identical copies of the same chromosome. So anytime you see these chromosomes lined up like this in the shape of an X, in actuality, in real life, they're really not crossed like that, but really touching at the middle and kind of bent. But when we're working with paper, I just show them in a cross. This is just one chromosome. It's two copies of that chromosome, but in terms of genetic information, we count it as just one chromosome. So this cell has 1, 2, 3, 4, 5, 6 chromosomes. In other words, its diploid number is 6. Now, I want you guys to think of each one of these letters as a gene that codes for a certain characteristic. So we could think maybe like A codes for the development of the head. Maybe B codes for the development of the forelimbs and the thorax. Perhaps gene C codes for the development of the eyes in a certain area. Okay? Each one of these letters codes for a certain aspect or developmental aspect of that organism. So when we make sperm or sex cells, we're going to need to make sure that all of the letters are present in each of the cells. If you end up with a sex cell that doesn't have G, H, and I, for example, then whatever organism gets fertilized by that sex cell isn't going to have a lot of the genes it needs to develop its proper structures. So for example, if G, H, and I coded for the development of, say, the lungs, the heart, and the liver, then the organism wouldn't have any of those things if it's missing those genes. All right, so today you guys underwent meiosis. So, as we learned in meiosis, the homologous pairs line up across from each other, and they split. So that we end up with two daughter cells, and both of these daughter cells, notice, have a complete set of genes. A through I. But they have different versions. For example, this daughter cell gets I star, whereas this daughter cell gets I. At this point, these two cells are haploid, because if you notice, these are just identical copies of the same chromosome. So they just have A, no A star. Just have B star, no B. Right? So two haploid cells, and they are different. Okay, we'll go through our second round of cell division. And we've got a cell with these genes, a cell with these genes, and this cell is going to divide as well. Giving us this gene set, right? So here are our 4 cells. So today in class, you were asked to write down the genotype of each of these cells. In other words, which alleles for each of the genes, A through I, the cells contain. So, why is this important? Well, let's look at the genotype for this cell. A, B star, C star, D star, E star, F star, G star, H, and I star. Notice that it is exactly the same as this cell, okay? So these are two sperm cells that this parent individual might have produced, right? Now, this one has a couple slightly different variants in it than these two sperm cells. So for example, A, B, C, D, E, F star, G, H star, and I. Notice that this sperm cell gets B star, whereas this sperm cell gets B. So both of these genes code for the same information. I think we said earlier that they code for the development of legs in the thorax. But this is going to be a slightly mutated version of the gene. So the development of the legs is going to be slightly different in this organism. Maybe this organism is going to have slightly longer legs, okay? This one is going to have slightly shorter ones, okay? So the importance of all this is there are two possible types of sperm cells, these two or these two, right? So let's say that it's this one that ends up going and fertilizing the egg. The contribution of this parent would be to tell the offspring to have longer legs. If it was this sperm cell that fertilized the egg, it would be that the offspring has shorter legs. So this really helps answer the question, why do siblings look different? Okay? They come from the same parents with the same genes. The answer is that parents contribute different combinations of their genes depending on what types of sperm or egg cells they produce. Right? So the key message here is that meiosis creates variation. And that'll take us to part 2, which is explaining the homework that you're going to do for tonight. So let's reset. So here we are back where we started, right? Same parent cell, same genome. The question now is, can we create sperm or egg cells with different combinations of alleles than that which we saw in the last example? So we saw, for example, that this setup creates two different types of sperm/egg cells, all right? One thing you're going to try for homework tonight in part 3 is doing the same setup, but then just switching which side two of the chromosomes line up on. So you're going to do something like that, and you could pick any of the chromosomes with which to do that. You could pick this pair or even this pair. And you're going to see if the sex cells, the 4 cells that are produced by meiosis, are different from what you had gotten the previous time. That's part 3. So finally, for part 4, you're going to model a process called crossing over. Crossing over occurs in the first metaphase of meiosis, and it happens when these homologous pairs are touching slightly. Now, when these homologous pairs touch slightly like this, they have the potential to break off and combine with the other set of chromosomes. So the way you're going to do this is simple. You're simply going to pick 2 that are kind of touching, and you're going to rip them off, like that. So we've ripped them off. Then you are going to take some tape, switch the 2 sides, tape them on. So this G came from this chromosome, ripped off, and crossed over to the other side. And you're just going to use some tape to attach it to the other chromosome. Repeat that process with this side, tape it on, and then reset it back to metaphase. Now from here, you'll proceed through meiosis again. So once again, first division, show the 2 daughter cells. You guys get the idea. Second division. This closes in. You get the 2 daughter cells. Two daughter cells on this side, and 2 daughter cells on this side. And from here, you'll complete the worksheet saying the genotype of each of these 4 cells. So that does it. And remember, the thing to keep in mind for tonight's homework are how meiosis can produce very different types of sex cells, all from one single parent, okay? And this helps you answer the question, why are siblings different from each other? Thank you and good luck.